This almost never happened. CRISPR gene editing. In 2011, two scientists met for the first time at a café in Puerto Rico. One was Emmanuelle Charpentier, a microbiologist studying a strange defense system inside bacteria. The other was Jennifer Doudna, an expert in RNA. They had both been invited to the same scientific conference, and a colleague simply decided to introduce them. That casual introduction would help create one of the most powerful technologies in human history. Because today, CRISPR allows scientists to cut and modify DNA almost as if they were editing a sentence on a computer. And it all began with bacteria. For billions of years, bacteria have been attacked by viruses. When one of these viruses enters a bacterial cell, it injects its own DNA and forces the bacterium to create more viruses. But some bacteria survive. They capture a tiny piece of the virus's DNA and store it inside their own genetic code. These stored fragments act like a collection of wanted posters. If the same virus attacks again, the bacterium creates a matching piece of RNA, which searches for the viral DNA. Once it finds a match, a protein called Cas9 cuts the virus's DNA and stops the infection. This natural defense system is called CRISPR. Scientists first noticed its strange repeating DNA patterns in bacteria in 1987. But for years, no one understood what they were for. Spanish microbiologist Francisco Mojica spent much of the 1990s studying these mysterious sequences in salt-loving microorganisms. Eventually, he realized that the unusual DNA between the repeated sections matched DNA from viruses. Mojica proposed that bacteria were saving pieces of their enemies and using them as a genetic immune system. His idea was so new that his paper was rejected by several major scientific journals before finally being published in 2005. The discovery was important, but CRISPR was still only something bacteria used to defend themselves. That changed after Charpentier began studying one particular CRISPR system. Her research showed that it relied on Cas9 and two small pieces of RNA. She suspected that this system could be simplified, but she needed someone who deeply understood RNA molecules. Then came the meeting in Puerto Rico. After the conference, Charpentier and Doudna walked through the streets of Old San Juan, discussing Cas9 and the possibility of working together. Their laboratories were on different continents, but their teams began collaborating. They combined the two natural RNA molecules into a single guide RNA. By changing the sequence of that guide, they could direct Cas9 toward a chosen piece of DNA. The guide RNA worked like a GPS coordinate. Cas9 worked like molecular scissors. In 2012, their team demonstrated that this system could be programmed to cut DNA at specific locations. Soon afterward, other researchers showed that it could edit genes inside living cells. Before CRISPR, changing DNA was possible, but it was often slow, difficult, and expensive. CRISPR made gene editing dramatically simpler and more accessible. A researcher could design a guide RNA, send Cas9 to a target gene, cut it, and then use the cell's natural repair process to disable, remove, or replace genetic material. Suddenly, scientists could study what individual genes actually do. They could create crops that resist disease, investigate new cancer treatments, and begin developing therapies for inherited disorders. In 2020, Charpentier and Doudna received the Nobel Prize in Chemistry. 3 years later, the United States approved the first treatment using CRISPR. The therapy, called Casgevy, edits a patient's own blood stem cells to treat sickle cell disease, a painful condition caused by a single genetic mutation. But the same power that makes CRISPR promising also makes it dangerous. Editing cells in one patient is very different from editing embryos, because changes made to an embryo could be inherited by future generations. A mistake would not necessarily end with one person. It could enter the human gene pool. There are also questions about unintended edits, unequal access, and the possibility of using the technology not only to prevent disease, but to select traits such as appearance, strength, or intelligence. CRISPR is not a magic wand. It is a tool. And like every powerful tool, its impact depends on who controls it, how carefully it is used, and where society decides to draw the line. Still, think about how close this story came to unfolding differently. For years, CRISPR looked like nothing more than a strange pattern inside bacteria. Mojica's explanation struggled to get published, and the collaboration that transformed it into a programmable gene editor began because two scientists happened to attend the same conference and were introduced at a café. A microscopic defense system, hidden inside bacteria for billions of years, may now allow humanity to rewrite the code of life.